And today we have two papers from, well, actually today's date, and we have one paper from one of the previous seminars. And we do not have actually one person who is going to participate in two talks for today, but I think it's fine that we start with our first talk, and then maybe he joins us a bit later. So if Ilya and Pavel are ready, you could start with the talk on paper about this.
Can you hear me?
Yeah, sure.
It's okay. Just and now my screen.
Yes.
Today we are going to present you Beast. Beast is Bayesian evaluation analysis sampling tool. Today in the modern molecular analysis, understanding sequences variation is a crucial moment, and this understanding is deeply rooted in statistical analysis. It's widely accepted that molecular sequence questions are essentially statistical. So involving parameter estimation and hypothesis testing. So accurate models that describe molecular sequences variation must also incorporate an evolutionary perspective. So BEAST addresses these needs by providing a robust framework for parameter estimation and hypothesis testing, making it essential tool for researchers studying the evolutionary relationship between sequences. BEAST employs a Bayesian statistical framework, specifically Bayesian Markov chain Monte Carlo model, or MCMC. This approach is particularly powerful because it allows researchers to implement highly complex evolutionary models. Bayesian MCMC provides a robust way to estimate parameters and test hypotheses. This method has been widely adopted in phylogenetic reconstruction and other evolutionary analysis due to its efficiency and the depth of insights it provides. So one of the standout features of BEAST is it focused on phylogenesis and genealogies. This means that BEAST incorporates a time scale into rooted trees, providing a temporal context to evolutionary relationships. Another significant feature is support for relaxed molecular clock models, which do not assume a constant rate of evolution across all lineages. This flexibility allows for a more accurate and realistic depiction of evolutionary processes, accommodating varying rates of change across different branches of the tree. BEAST is designed to integrate a wide range of evolutionary models into a single framework. These models include substitution models, insertion deletion models, and also demographic models, tree-shape priors, relaxed clock models, and node calibration models. This integration allows for construction of complex evolutionary models from simpler components, providing a more nuanced and detailed analysis of molecular sequences. By avoiding unnecessary simplifying assumptions, BEAST can offer a more accurate and comprehensive understanding of evolutionary process. So, Ilya, please.
Yeah, thank you. Can you hear me?
Yeah, sure.
Okay. So BEAST offers flexible options in defining the evolutionary model for genetic analysis, such as accounting variant substitution rates and heterogeneity among sites and branches, while maintaining a consistent ratio of transitions to transversions. All parameters, including the tree structure, can be shared or independent between partitions of the sequence data. Bayesian analysis in BEAST requires prior distributions for parameter values, which allows researchers to incorporate relevant knowledge or impose informative priors. Different types of priors like exponential, normal, log normal, or uniform can be assigned, which enables the specification of specific mean values for parameters like the age of the tree's root. The evolutionary model for a set of aligned nucleotide or amino acid sequences in BEAST is divided into 5 components. For each of these, a range of possibilities are offered, and thus a large number of unique evolutionary models can be easily constructed. And these components are substitution model. This is a homogeneous Markov process that defines the relative rates at which different substitutions occur along a branch in the tree. Rate model among sites. The rate model among sites defines the distribution of relative rates of evolutionary change among sites. Rate model among branches. This model defines the distribution of rates among branches, and it's used to convert the tree, which is in units of time, to units of substitutions. These models are important for divergence time estimation procedures. Tree. This is a model of the phylogenetic or genealogical relationships of the sequences. And tree prior, this model provides a parameterized prior distribution for the node heights in units of time and tree topology. Let's take a closer look at these components. Substitution models help us to understand how substitutions happen. Various substitution model can be used for nucleotide and amino acid data, including GTR, HK85, Blossom, etc. For coding sequences, the Goldman and Young model is applicable for codon evolution rate among sites can be described using gamma describe rates proportion of invariant sites of a combination of both. Sorry, wait a minute, please. Yes, rate models among branches. These models help scientists to understand and describe how different species are related. The basic model for rates supported by BEAST is the strict molecular clock model, which is calibrated by specifying a substitution rate or a nodes rate. This allows the estimation of divergence of dates for specific clades. Clades can be defined by enforced grouping or as the most recent common ancestor of a set of taxa. The model can incorporate defines differences in tip dates to provide information about the overall rate of evolutionary change. BEAST also includes relaxed phylogenetic models allowing for variable rates of evolution among branches. The evolutionary rate must be specified when sequencing data is from one time point. This rate determines the units of node heights, demographic parameters, and population size. Sorry. The units can be mutations per site per year, or mutations per site per generation. The resulting tree will be in units of years, generations, or natural units, depending on the specified evolutionary tree. Tree priors, like starting points of guidance that help scientists to construct phylogenetic trees. They provide a framework for understanding the evolutionary relationships between species, taking into account different factors like population genetics, speciation rates, like yellow process priors, and the balance of species, Bessus and deaths. This is like both death process priors. These priors are essential for making accurate evolutionary trees, especially when dealing with incomplete or uncertain data. Finally, BEAST also has a model of the insertion and deletion process. BEAST allows for the analysis of multiple data partitions simultaneously. It's useful for combining multiple genes in a coalescing analysis or assigning different evolutionary processes to different parts of sequence alignment. Parameters such as substitution model, rate model, tree structure, and tree prior can be linked or unlinked in the analysis. Next slide, please. Model comparison and model selection. When comparing different models in Bayesian framework, the goal is to determine which model better explains the data. The most rigorous method is to achieve this through calculation of the Bayes factor. And the Bayes factor is a ratio that compares the marginal likelihood of two models. This is the probability of the data given a particular model averaged over all possible parameter values of that model. So the Bayesian factor for comparing two models M1 and M2 is given by the probability of the data D given model M1 divided by the probability of the data D given model 2, where the probability of the data D given model M is the marginal likelihood of model M averaged over the model parameter values data. Specifically, the probability of the data D given the model M is equal to the integral of the probability of the data D given the parameter values theta and model M times the probability of the parameter values theta given the model M with respect to theta. Calculating Bayesian factor typically involves reversible jump Markov chain Monte Carlo, which is not yet implemented in BEAST. However, the alternative methods to for approximating the marginal likelihood, such as importance sampling with the posterior as the importance distribution. The harmonic mean of sampled likelihoods can be used as an estimator of the marginal likelihood. You can see this equation at number 3. This estimator doesn't always behave very well, but there are number of modifications that can be used to stabilize, and bootstrapping can be employed to assess the uncertainty in the estimated marginal likelihoods. In general, Bayesian factor more than 20 is a strong support for the favored model. Next slide, please. Here you can see an example where we analyzed 17 sequences of dengue virus serotype 4 from 1956 to 1994 using BEAST under both a codon-specific codon position-specific model, GTR plus CP, and a standard model, GTR plus gamma plus I. Both of these models have the same number of the parameters. Strict and relaxed clock models, assuming constant population size, were tested. Each analysis involved 10 million Markov chain Monte Carlo steps with substantial effective sample sizes. The GTR plus CP model fit the data better than the GTR plus gamma plus I model and supported a different tree topology. Little differences between strict and relaxed clocks, suggesting clock-like evolution, was found. Next slide, please. The root date was estimated around 1924, with a substitution rate of approximately 8.38 times 10 to the power of -4 per year. The GTR plus CP models produced more precise and concentrated posterior distributions of tree topologies compared to GTR plus gamma plus I models. Pavel, please continue.
So, BEAST is several critical components that work together to facilitate evolutionary analysis. One of them, Beauty, is a graphical UI that allows users to generate XML input files for Beast. This interface simplifies the process of setting up analysis by providing a user-friendly way to specify data and models. Tracer is another important component that's designed for the analysis of MCMC output. It helps researchers visualize and interpret the results of their analysis, make it easier to assess the performance and accuracy of the models. The core program of Beast processes the XML input files and produces text file outputs, summarizing the estimated posterior distributions of parameter values and trees. Beast operates through a methodology that begins with XML file, as I said. This file details the data to be analyzed, the models to be used, and the technical parameters of the MC algorithm, such as a proposal distribution, chain length, and output options. The output of a Beast analysis is a set of plain text files that provide estimates of the posterior distribution of parameter values and trees. This format facilitates reproducibility, allowing the researchers to replicate and verify the analysis. Additionally, it supports detailed examination of the results, ensuring robust and reliable evolutionary inferences. So looking ahead, the development of Beast continues to introduce some new enhancements. Future versions of Beast will include birth date priors for tree shape, which will improve the modeling of the speciation and extinction processes. There will be also faster and more flexible codon-based substitution models, enhancing the analysis of sequence evolution and nucleotide level. The structured coalescent will allow for a modeling of subdivided populations with migration, providing a more detailed understanding of population dynamics. Models of continuous character evolution will enable the analysis of traits that change gradually over time. Lastly, relaxed clock models based on random local molecular clocks will offer even greater flexibility in accounting for varying rates of evolution. In conclusion, Beast stands out as a flexible and comprehensive package for evolutionary analysis by combining multiple models into a single framework. So Beast offers unparalleled depth and accuracy in the study of molecular sequences. The ongoing development and introduction of new features ensure that Beast remains at the forefront of evolution in bioinformatics. Its ability to integrate complex models and provide detailed analysis makes it an indispensable tool for researchers in the field, continually pushing the boundaries of what we can understand about evolutionary process. So that's all, and thank you for your attention.
All right, thank you. So are there any questions from the audience? Oh, I have a small question. So have you looked at whether or not this still supported, this tool? Is it regularly updated, or how modern it is? Is it used right now? I mean,
I'm not sure, but for my research work, I'll use the Beast. I will use the Beast in some time. So I think I'll notice that moment, but now I can't say it. Anyway, this tool looks just quite new comparing to others in this field.
Well, yeah, I think it is regularly supported even right now, but it's not like the modern version is not the first one. I think they switched to Beast 2 at some point. So yeah, the original tool is not, well, it is supported, but not really as much as the second version. All right, so maybe any further questions? Or maybe not. So if not, so thank you. And
Thank you.
yeah, let's now go. Well, we should probably go to our second and third talks, but we do not have right now a person with us that should present both of these papers. So do you have any way to contact him, perhaps?
Who do we need to?
Denis Novoseltsov.
I don't know where is he, but I can tell you like 90% of the topic if you need.
Oh, so did he even participate? I mean, well,
Yeah, of course, but we dive deep into the theme together, so we just helped each other with the theme.
Okay, because I thought maybe he dropped the studies at some point. Maybe he didn't even participate at all. But if
No.
he doesn't, oh, all right. I guess it is fine for if we do not have any other options. If he's not coming, I guess you could
Okay.
present by yourself.
Yeah, just a moment. Yeah, I will start from the very beginning. Do you hear me well, by the way?
Yeah, sure.
I will start from the phylogenetics itself. Maybe somebody needs to refresh the information in their mind. So we will be talking about building trees. Actually, it's just a topological or mathematical analogy. That's the way we present the information about different clades, about different species and their genomes or proteins sequences. So even if we change some like edges of the tree or taxons, they stay the same sometimes, just like on the right picture. There are two trees and basically that's the same tree. So we can build a tree in different ways. I think everybody knows that already, and they can be unrooted, they can be rooted, for example, they can be weighted or unweighted, and they are used everywhere. Like in biology, they are very popular. So how should we build them if we want to? At first, we need sequence alignment. So we need to see are the sequences are even like from the same clade and how they differ. We need to put them on each under another, one under another. Okay, now we have an alignment. How do we know exactly some numbers? How can we estimate the difference between them? Actually, it's not so easy. You can just count the differences like 1, 2, 3. We have different models for that because evolution and mutational processes are sometimes tricky, and we can do it in different ways. For example, we can estimate compositional change, or we can do some Markov models, etc., etc. Okay, when we estimated the difference, we need an algorithm that can build a tree by itself. They can be easy, they can be difficult, and they can be very different. And after we build a tree, we need to evaluate it. So did we build the perfect tree? Is it okay? Can we build another one? And how to
Well, it seems like you suddenly stopped talking. Maybe your, well, microphone stopped.
sorry, is it okay now?
Yeah, it is all right.
Sorry. So let's go back to tree building. There are two groups of methods. Distance-based methods are kind of older, and they are just building a matrix of distances. For example, they are comparing each leaf of the tree to another leaf. They are counting different differences and putting it into a matrix. And each step, they are making it matrix shorter and shorter, and finally we have a tree. Character-based methods, they are not about distance, but also about probabilities. They are trying to count probabilities of every single tree. So, for example, substitution of each nucleotide or each amino acid can be different. So, for example, G can become C way more often, for example, and different organisms might have different probabilities about changing nucleotides, and different mathematicians can make different models. For example, I think there are hundreds of them already, so it might be important if you want to get a better tree, a most probable one, and the closest to reality. But if you want to count the probability of every single tree, can we actually do that? Because sometimes we have so many species, so many sequences that we will put days and hours into the process, and we will simply run out of our CPU. So how can we do it faster? This problem can be solved by IQ-tree. It's a pretty simple application, like user-friendly. It can be used in terminal, simply just one line. You can put different flags and use it simply. It implements such algorithms as model finder or Bootstrap, for example, so it has everything in it. Also, you can use it online. That's the interface. It's pretty understandable, so you can try it there if you want. So what's the point? What is behind this algorithm? Well, at first, we can make random trees. For example, 100 trees, right? Count the probabilities of them. They will be very different. And if we find the best one, we can just make it as template, and then we can take a tree and change the branches like it is shown on the right. For example, on this tree, in the corner, we can make 2 global changes, right? Or 3, yeah. And we will get like 6 trees. But the base is the same, so we can choose the best base at first and then swap the branches as we want to get the best one at the end. It will take much less time and resources. So this algorithm is called nearest neighbor interchange. That's the IQ tree in the nutshell. So yeah, it generates 100 parsimony trees using RxML and chooses the best one, top 20 trees. Okay. What about different models? I've told about Model Finder already. It's the application that is implemented into IQ-tree, and it helps you to choose the best substitutional models to count these probabilities of change in nucleotides or amino acids. And there are lots of them. They can be mathematically based on different species, on different populations, and they can be counted in different ways. So sometimes it's not obvious how to choose the right one. If you are not really familiar with them, it's better to just put it on auto mode and just use Model Finder. It will do all the work for you. Then you can use this search tree algorithm and, for example, use Bootstrap. There are also a few other algorithms to to choose the best tree, to tell that it's the best really, and IQ-tree will help with it you as well. Bootstrap is the thing where you generate like a lot of trees and choose the most popular one, for example. So you are just repeating the algorithm again and again. So IQ-tree was compared with the other applications which are building trees the same way based on probabilities, and it appeared to be the same efficiency as the most popular RxML already and PhyML. Yeah, almost the same efficiency as I can see. So as it has solved a few problems already, and it takes the same time to count data, you can use it, and IQ-tree became really popular these days. So that's basically it, I think. Sorry for missing one slide. I'm not really familiar with the deep algorithm, deep math behind it, but yeah.
All right, it is fine.
Mm-hmm.
So any questions from any other people? So, well, probably there are none. So thank you for your presentation. And let's now go ahead and continue with our final talk. And it's going to be, well, ideally it should have included Denise as well, but given the circumstances, I'm going to ask Daniil if he can do it by himself.
Yeah.
Right,
so
sure.
just a second. Yeah. Okay. Hello everybody, colleagues. Today I'm going to talk to you about the metagenomics topic. I suddenly missed it. So it is about the kraken, one of the most popular metagenomic sequence classificator behind the days. And now its second version is still very, very popular. So, first of all, really briefly, how modern metagenomics works. We have samples. In samples, we have environment and DNA, or it could be any other nucleotide, possibly. Nowadays, we can talk about metatranscriptomics, and sometimes really crazy people do even meta chipsex or something like that. But it is not the topic now. And we have a nucleotide of environmental some environmental nucleotides, and we can sequence them to get a metagenome or transcriptome or something else. By analyzing it, we can find the network between some species, and then we can create some predictions about differences between samples. We can generate different models, including machine learning models based on that. We can predict different diseases using modern metagenomics, and a lot other interesting things. But always one of the most important ideas of metagenomics is to just explore microbiome. And to do it, we try to classify the metagenome in a nutshell and try to measure accuracy of the prediction. How many species are in the metagenome, and some related questions about blood distributions and so on. So we have a different approach in metagenome annotations. And first of all, of course, there are two really big parts of the metagenome of modern metagenomics. Sensillata, if we talk about it, we could include their amplicon sequencing. Nowadays, I believe that more than 90% of the articles use Chime, the second version of it, to analyze amplicon data. But if we talk about the metagenomic sensu stricta, also known as shotgun metagenomics, where we sequence all possible DNA from environment or just exclude RNA sometimes, ribosomal RNA genes, yes, of course, so to get the classification. We have pretty different approaches. If we assemble metagenome, we could predict assembled parts of it using Metabot, the second version of it now, and some other classifiers. Also, we of course could use for it Kraken or other camera tools, but it is not very efficient. For assembling of the metagenome is a pretty tricky part. So for big metagenomes, it is really time consuming. So really a lot of different articles don't use assembling for metagenomes and just classify raw reads. To do this step, we can use two different approaches. We could just do a basic search of something, or we can do camera search. Nowadays, only, as I know, only really distributed program that use camera search is Kraken and other its versions like Kraken Unique, Kraken the Second, and so on. And other approach is a basic search where we can choose between searching on the full database, which different BLAST implications work. And we can search through the marker database. That is how Kaiju and Metaflan works. And now let's talk about Kraken, how it works in a nutshell. First of all, it generates a database. And in compared to the full database of nucleotide, Kraken database contains full nucleotide database on the RefSeq, and Sabay database is nearly 600 of gigabytes. And if we measure Kraken database, it is only 30, from 30 to 60 gigabytes, so it is much smaller. And also there is a mini Kraken version which requires only 4 gigabytes for the database. how it works? It predicts k-mer for each sequence in a reference database, and then it generates all possible distributed k-mers, and then minimize their abundance based on the jellyfish counter. It is the most tricky part of its installation, just to tell Kraken where jellyfish is, and almost always it is broken on this stage, but still it is really popular. And the duration of the Kraken database, it is a really long stay process. Comparing to, but there is a lot of distributions or distributors together the pre-built Kraken database. It is not a problem if you wouldn't use the custom database. Why should you use custom database? We will talk about later on. And there is not only the building the database is a long process, also putting it into physical memory is a long process. So if you can use Kraken on a lot of samples, it is really faster to not to do it in the loop, but to use a list comprehension or something like that. It uses the default k-mer length of 31. It is pretty same to basic BLAST approaches. For example, MegaBLAST use 28 k-mer length. Kraken has a uniform. Kraken database has a uniform format with other tools, except of Kraken the second version, and a really big part of the higher sensitivity of the second Kraken version is the slightly another database. How the prediction algorithm works? First of all, k-mers are aligned to a sequence, and then, of course, not a line, only searched, and then it is a voting algorithm. We have of course different k-mers could belong to different taxonomy parts, and if we have a common k-mer for a really big part of the phylogenetic tree, we wouldn't like to classify it on such a top level. So when we have a really big number of k-mer across all the sequence, we would like to classify it not in exact way, but in a probability way. It's where the voting starts, and the second version of Kraken really improves this part. Here we when we use the first version of Kraken, almost always if we have any outlier in a query sequence, so for example, we have a mutation like copy variation, and we have a really long sequence. First version of Kraken predicts that if it predicts that this copy variation is from the really different organism, or it just don't know how to measure it, it just predicts really high level classification. It is a pretty common problem for any classification tool, but for Kraken, it also but for Kraken approach, it is a really common problem. And for example, different classifiers that used just a second, like in the flan, yeah, they just use the model database with only very well-known sequences to predict something. It just evaluate really good the known species, and all unknown species it predict really, really bad, but just mark them as unclassified. While Kraken try to classify it really deeply and and needs really big thresholds to don't make the classification just a nightmare or the noise, because it predict for any problem it just try to predict something like really, really different species. For example, Metaplan on a human gut sample predicts nearly 200 species, while Kraken try to predict more than 2,000 species, with most of them have the abundance less than 1%, much less than 1%. So on the one hand, it is a big problem, but on the other hand, it is a really, really good strategy for different machine learning tools, because we have a lot of different genomic signatures that could be classified in any way. But if they are classified, if we really don't want to evaluate how good this classification is, Kraken is, I believe, the best tool to do this classification step. In the article, they evaluate the sensitivity and precision of Kraken. First of all, they evaluate it in comparison to other tools of that days. It was there are 5 tools in comparison: Kraken, Megablast, and Fimbel and MBC. It is less common tools nowadays, and Metaflow. Kraken have a is a really, really fast approach, but it's worth mentioning that this prediction was done using really, really a lot of reads. I believe it is more than 10 billions using in the article to evaluate counts per minute. But as I have mentioned, just putting it into a memory its database is a really long process. And also, if you haven't run space enough, it is much, much slower. On this charts, there are 2 main axes. It is sensitivity and precision. Sensitivity here is something like the pole, so it is just how many species are classified right, and in comparison to all classified species. So we see that all these tools on a generated on just generated metagenome predicts less than 80% of all sequences correctly. And it is a common problem for any generated sequence exams, because if any bacteria in this example have something like not really regular DNA parts, like a lot of adenine and something like that, it just couldn't be predicted very well using pretty any approach. A similar situation is with the MySeq generated reads, and really, really different is with the simulated approach based on really a lot of bacteria in here. with much higher abundancy of the common species. So we see there that the gene sensitivity is really high. On the other hand, kraken, as well as metaflan, as well as megablast, are really precise. So their classifications would be really well used for different machine learning approaches. Secondly, they make faster versions and smaller versions of kraken. Kraken Q, mini kraken Q. And it is interesting that even on small databases, kraken predict species really well. It is interesting that the sensitivity, when sensitivity is higher, the precision is smaller. It is common problem when we evaluate different thresholds. And here we can see it on the high-sec data, but not on MySec as well as CMBA data. It is a really interesting topic about any chimera classifier of metagenomes. The usage of the curated database to predict species abilities. Why? Because when we have really big database, we have to deal with multi-classifiers parts and multi-alignment search isn't so having such a big distribution of the phylogenetic bias. So if you can use custom database for kraken, just use it. Also, they try to exclude some ranks to predict how well kraken classifies without them, and how precise is it. And it is pretty precise. Voting algorithm is really good here. That's basically it about the article, but I just like to tell something about why kraken is so popular nowadays. Because, as for me, it is a really interesting example of really big ecosystem around the tool, which really makes it shine. First of all, there is really big tool named Kraken Tools, and for example, base article about Kraken and its second version is just a BMC Genome Biology journal with impact nearly 10, while the Kraken Tools has own article in Nature Protocols with much higher impact. Kraken tools could be used for extract data from Kraken annotations. For example, one of the most common and the biggest problems of any metagenome annotation is a host genome prediction in a metagenome. It is most often there are more than 90% of sequences in any host-related genome, which are related to host. So we want to remove them before any usage of other tools on this metagenome. So it is much better to use different dehostification programs, and one common approach is just the Kraken tools. Another example is Corona charts. In the article, they made one of them, and now it is a separate program which also can use other classifiers like metaflan to draw pretty beautiful charts. it is also interesting that on the simulated data based on real metagenomic sequencing, about 60% of all reads were unclassified using kraken. And of them, only 11% would be classified to any rank using megablast. So what is this unclassified abundance? We really don't know. Most of researchers tell that it should be viral. Personally, I think that most of it should be different fungal species because kraken as well as megablast have really big troubles with the fungal abundance classifier, while other tools which use only marker genes like Kaiju or Metaflan are much better for it. So here we can see only bacterial abundance. Another common tool is a Phylogenetics tool, which generates really beautiful reports for Kraken. I by myself write the tool called Architectis, which also describes the data in a Kraken reports, but in other ways. And now, the most common tool for analysis in metagenomics, I believe, is the Metaflan, the fourth and the second version of Kraken. It is much faster than first version. It is a little bit more precise, and it has also bigger sensitivity. The golden standard of analyzing one of the golden standards of analyzing metagenomics data in modern era is usage of second version of Kraken with Bracken for classifier, which really improves the output. So I believe that should be it. So Kraken is really good program which have really big impact on modern metagenomics. It's a conversion on the golden standard. Unlike other classifiers, it use just a chimer classifier. It is really good for classification of most bacterial taxa, but it just, I believe, breaks when you try to use Kraken for when you use Kraken on short reads with trying to classify eukaryotic or viral genomes. Basically, that's it. Thank you very much. It was very long.
All right, thank you for a very comprehensive review of the Kraken and everything around it. So, any questions from your colleagues?
Yeah, may I?
Yeah, sure.
Well, if at first, for example, I use Kraken 2 and I'm left with some unclassified reads, what should I do next? For example, I've ran two tools, Kraken 2 or Metaflan 2. I've got two results. How can I combine them in order to get rid of the most of unclassified reads? Can I just combine them somehow?
Yeah, it is really interesting question. Nowadays, I try to develop my own tool for it. It is a part of the Architectis reports. But basically, you just couldn't compare it in a nutshell because they use different approaches. And one, of course, always classified less than Kraken. So you can't combine them in a nutshell. Also, they have a slightly different reports, which is just a nightmare to always compare them. But basically, you, if you really want, you can use different classifications as a different inputs for your other classifier, or your sample classifier. or just to evaluate the difference and predict comparisons between two different tools and try to measure the difference between them. It is the most basic approach. Most of articles that I have read, if they use different classifiers, they just try to compare the results, but not to combine them. It is a really big problem now. And
Got it.
about the second question you have got, I got it. It was about unclassified, yeah?
Yeah, if I have a lot of unclassified reads, how to properly analyze them, especially if they were previously paired, but now they are unpaired and it's a great problem for some tools.
I believe that the second version of Kraken can get output also in I know that Kraken tools can get the output in paired reads as well, so it isn't really a big problem. But to deal with unclassified, you can basically do two different approaches. First of all, you can try to blast them in more time, but in most metagenome samples, it wouldn't really help. It can help only if you have really, really long reads, like if you have nanopore sequencing data, or if you have MySync data. But if you have, but still in metagenomes, you often have more than 80% of unclassified in output of Kraken or other programs. And BLAST can classify it only about 10 to 20%. All other reads in most of articles are believed to be just a noise or just something that we can't classify nowadays. But other articles try to measure the connectivity between the amounts of these unclassified reads in connection to classified, and try to build the network of the genes based on just the correlation of sequence abundances of classified and unclassified reads. And then if the correlation is really, really strong, just to classify them based on this correlation. It isn't really a good approach. I haven't seen any really good program about it, only a lot of different custom scripts. But it is, but using this approach, if you have a lot of samples, you just need more than at least 20 samples to do it. And better, really better to much.
Have you seen a tool called Agnostos?
I believe yes, but a long time ago, so it is
Yeah,
interesting
it's like
to see now.
I thought maybe it was kind of relevant that it is a tool specifically for uncharacterized sequences that usually they're thrown away, but it is a part of an Anvil toolbox. You probably have heard of it.
Yeah, it is common for me. Yeah, I know, but as for me, it wouldn't work very well. It just tried to classify it, unclassified abundances, but it just broke because I have.
All right.
I believe not very lot of samples.
All right. So I've just heard about it. I haven't used it for its purpose. I was just saying that I read about it. I read the paper about it. I like the idea, but I haven't tried it myself. So that may be an answer also to Anna's question about what to do with these unknown, unclassified sequences.
Yeah,
it is
thanks,
a really
but
big problem.
it sounds very sounds like an heuristic program, so it's not the perfect algorithm, I guess.
Yes, of course, and I believe that the best approach is to just write a script by yourself and try to evaluate real distributions of the samples and just compare these distributions. I believe that's the only way is to classify it. It's just a usage of the network approach. Yeah. So I don't know to do this.
Maybe we can also combine different libraries of Kraken. Can we just put it in one line in Kraken and like use it at the same time, different libraries?
I believe that is not really the case any times, because if you really want to use different libraries, you just can develop library by yourself, and it is not a problem. It is just a subprogram of the Kraken, which is called Kraken build. Usage it's if you have installed Jellyfish correctly, it is just really simple, but time consuming, of course. I have done a lot of comparisons between different Kraken Kraken second databases, and most always they get really similar results across the species if you use bacterial database or less database. If you try to use full database, which is called NC, as I remember, it is a nightmare. You just don't do it. If you wouldn't to do only machine learning steps, its predictions are really bad. You will have really a lot of species that just couldn't be there anytime. So combining and it still wouldn't happen help to classify the unclassified amount.
All right. Well, thank you once again for your wonderful presentation. And I guess that's it for today also. So thank you all for coming. And with that, goodbye, everyone. Have a nice day.
Thank you.
Thank you.
Goodbye.
Thank you. Goodbye.